G protein coupled receptors. The example of epinephrine. The epinephrine bonds to the G protein coupled receptor, which brings over the G protein and turns it from GDP to GTP. The GTP goes and deposits the third phosphate at the adenylyl cyclase, and the adenylyl cyclase pushes the phosphate and it turns to CAMP. The CAMP activates the regulatory portion of an enzyme, and the catalytic portion receives an ATP and activates phosphorylase, which then releases glucose to the bloodstream. Receptor tyrosine kinase protein. A ligand bonds to receptor tyrosine kinase protein, which makes it into a dimer, and the dimer takes 6 phosphates and gives it to a relay protein, turns it to an activated protein, which then changes the cell response. The normal insulin transduction pathway, receptor tyrosine kinase protein, which receives insulin and changes to a dimer, releases 6 phosphates into the IRS1, which then activates P3 kinase, which changes PIP2 to PIP3. The P10 up there can change it back, which activates AKT, which tells the cell to put GLUT4 in the membrane, which takes glucose from the bloodstream into the cell, and AKT also tells the cell to synthesize glycogen. In diabetes, insulin is taken into the insulin receptor, but no cell changes happen, and GLUT4 is not used, and there's no glycogen synthesis happening. So what happens is insulin is not changing anything in the cell, and so because of that, the glucose piles up in the bloodstream, and so does the insulin, and it results in high blood sugar.